Good.
Hello everyone, and thanks for joining us for the Spatial Biology Revolution 2024. I'm Heather James, and I'm here to moderate today's event alongside my colleagues George Hammond and Princess Sennett. Before we start the talks today, I just have some general information and a few tips for getting the most out of the event. We would like to ask if you could please refrain from video, audio, or photo recording of all talks. Many of our speakers have kindly agreed to share their slides with you, and these are available within the literature section on the platform. Please remember that we encourage debate and discussion and welcome all of you to use our networking services to introduce yourselves to other attendees, but we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. For more detailed instructions on how to make the most of the features available to you at this event, please navigate to the event home and watch the overview video at the top of the page, or visit the FAQ section in the left-hand menu. With that out of the way, I'm delighted to introduce our first talk, Spatial and Functional Organization of the Human Genome. Please join me in welcoming Dr. Stefan Schoenfelder, co-founder and chief scientific advisor at Enhanc3D Genomics. Dr. Schoenfelder completed his studies in molecular biology at the University of Heidelberg before joining the Barbara Ham Institute in Cambridge, UK. With many major contributions to spatial research, Dr. Schoenfelder co-founded Enhanc3D Genomics. He now splits his time between this and leading his research program. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. Without further ado, I will now hand over to Dr. Schoenfelder to begin his presentation.
Thank you very much, Heather, for the kind introduction and the invitation. As Heather mentioned, I have a double affiliation at the Barbara Institute, so I'm half academic, half working for a biotech that I've co-founded, Enhanc3D Genomics, and my talk will reflect this. So I've split it up into several parts. I want to start by introducing the 3D organization of the human genome and why it is important. I'll then talk about enhancers in development and disease. Before I go on to talk you through some biochemical methods to link enhancers to their target genes, and I'll focus on one that we've developed and that is now the core technology at Enhanc3D Genomics. Then I want to tell you how we use this technology to advance human healthcare, what our plans are in this space. And then I'll go on to talk about some of my academic work, so of my work. This is the functional annotation of enhancers in human pluripotent stem cells. This is unpublished work, hot off the press. And I'll end with a model for enhancer promoter specificity. So, functional organization of the genome in 3D. The question that I've been interested in and fascinated by for the last at least 15 years or so of my career is this: how remote enhancers, and I'll come to explain what those are in a minute, relay regulatory information to their target genes. It's been called one of the central mysteries of genome organization and function. And more specifically, you can break this, what I'm interested in, what this fascinating question entails, into two sub-questions. So first is, how do enhancers find their target genes in the 3D space of the nucleus? And related to that, how are enhancers prevented from activating non-target genes? Because when they do, they're powerful regulatory elements. When they do, this is sometimes called enhancer hijacking, enhancer adoption. This can lead to disease states. So I want to take a step back first and remind you maybe about how much DNA we have in our body. You have about 2 meters of DNA in each of your cells if you span it out end to end. And you have about 10 trillion cells with DNA in them. The most abundant cell type in your body is the erythrocytes. They are so specialized they have actually kicked out the DNA. But there's plenty of cells, about 10 trillion left, which have DNA. So that makes about 20 billion kilometers. That's enough to span around the Earth 500,000 times, 26,000 times to the moon and back, 66 times to the sun and back. There's an awful lot of DNA in your body. So 2 meters in each cell, and that is squashed into a nucleus. That's about 10 microns across, so much smaller than a human hair in diameter. It's only visible, of course, with specialized equipment, microscopes. And for a long time, it was considered that just squashing this DNA into such a confined space is merely a logistical problem. It just needed to be compacted to just fit into this tiny space. And it wasn't thought that there was any more organization to this than there is to a random bowl of spaghetti that's curled up. But over the last 20 years or so, we've come to realize that that's not actually true, and that on top of this enormous compaction that needs to happen to fit this 2 meters of DNA in a tiny space of a nucleus, the DNA is folded in a cell type specific manner. So in a brain cell, we fold it differently than it is in a liver cell, to a skin cell. And that is a key contributor to what makes these cells different, what makes them look different and fulfill different functions. And I'll go into this in more detail in a minute, but the way it does this, it brings together different elements of the genome. Remember, the genome is the same in all these cell types, but they're all different because they express different set of genes. And the 3D genome architecture is a very upstream step that brings together different regulatory elements, so genes and the switches that switch them on and off, for example, to communicate and interact. So I'll talk about this. I've mentioned enhancers in passing, but I'll talk a bit more about them. Enhancers are cell type specific gene regulatory elements, so they're depicted in these red boxes here. And that's in contrast that distinguishes them from gene promoters and genes, which are, with some exceptions, but by and large, they're positioned in the same genomic locations. Enhancers can be located in genes or upstream and downstream, so they function in a position and orientation independent manner. And we have relatively good means to locate enhancers in the human genome. They're not perfect, and that's the second part of my talk. The data from my own lab, but they're reasonably good. What's much more difficult is to know which gene an enhancer regulates. And this is it's a famous example in developmental biology. So this red box here in the LMBR1 gene marks the position of a LIMPAT specific enhancer, but it regulates none of the surrounding genes, but instead a gene that is located almost a megabase, so almost 1 million base pairs away, 850,000 base pairs away from these. And it does so by looping out and forming a direct physical contact with the promoter of this gene, so this chromatin loops. When you delete this enhancer experimentally, this is a LIMPAT specific, so enhancers are very cell type specific, developmental stage specific. You can see that the mice are born with severely truncated limbs. And interestingly, point mutations in the same enhancer in humans, where the orientation of this enhancer and the respective orientation of the surrounding genes is conserved, results in polydactyly, so malformations, developmental malformations of the limbs. And this defect that you see of the truncated limbs is remarkably specific, because sonic hedgehog, the gene, is a key regulator in many developmental processes. But what's affected here is specifically its expression during the development of the limb, not in the brain, not in the gut, or in other developing organs. The enhancers that regulate sonic hedgehog expression in these developing organs sit in this kind of gene desert here. This one is specific for LIMPAT development. So the key points I wanted to make about enhancers is they're cell type specific gene regulatory elements, key regulators of cell fate, and they convey regulatory information through these chromatin loops, so direct contacts with their target gene promoters. I have one more slide on this enhancer, because this is my favorite enhancer. These are the same pictures that you've seen in the previous slide. Interestingly, it's now known that Hemingway had a cat that also suffered from, it's probably not suffering that much, but it had polydactyly, so an abnormal number of digits. And it's found that that's also caused by a mutation in the sonic hedgehog LIMPAT specific enhancer. You could still, descendants of this cat still live in the Hemingway house in Florida, Key West, Florida. This is perhaps my favorite sonic hedgehog paper, and there is quite a few. So what Evgeny Kwon and colleagues have done here is they've knocked in this enhancer sequence from 17 different vertebrate species into the mouse locus. So they've replaced it with a human, a fish, python, and this is the center as knockout. It's kind of a negative control. And you can see that even in distantly related species like the fish, the sonic hedgehog enhancer can support a sonic hedgehog expression, that's shown in this staining here, and proper limb formation. But when it comes to a snake sequence, it looks pretty much like the knockout. Obviously, snakes don't have limbs. You can take the snake enhancer and put 17 base pairs. It misses 17 base pairs. You put these 17 base pairs back into this sequence from either the mouse or the human, and you can perfectly restore sonic hedgehog expression in the developing limb and limb formation. It's a really fascinating example of enhancer. This is one example of an enhancer, the enhancer mutation. So remember, enhancer mutations are different to gene mutations. A gene is then mutated in every cell in your body, and the effect would be apparent then. And enhancer is also mutated in every cell in your body, but the effects, because the enhancer will only be active in very specific cell types, will only be manifest in the specific tissues. And there are others. The PAC6 enhancer, for example, causes the absence of the iris. SOX9 enhancer mutations in there cause developmental syndrome called Pierre-Robin syndrome. And these all act over a long genomic distances, 150 kb to over a megabase here. I should say, I'll talk about enhancers that function via these direct contacts with their promoters, and that is the predominant model of how enhancers function. But there are cases in biology, and it's never, one size fits all is almost never true. There are cases, reported individual cases, I just want to disclose that here for completeness, where enhancers seem to function without this direct contact. So they sometimes referred to as the contactless enhancers. It's unclear how they function, but it's interesting. I believe this is probably minority of the cases, but nonetheless, they're interesting. The experiment that convinced me that I really wanted to work in this space is a milestone experiment from a paper from Gerd Robel's lab, Dengendal in Cell, 2002. And what they have shown is that you can use an experimental trick to force the interaction between an enhancer and the beta globin gene. This is the enhancer called locus control region. So it doesn't matter for this purpose, but you can force them to come together experimentally, and that alone activated beta globin transcription over 1,000-fold. So it shows that just the juxtaposition of an enhancer with its target promoter can induce transcription. And that for me is a beautiful experiment and convinced me that this is what I wanted to study. So what we realized that the methodology was lacking to capture these enhancer promoter contacts that occur. In this case, they're forced, this is an experimental trick, but they occur all the time in nature, in cells. And the methodology to capture those was lacking. And this is what I set out to establish, and that's what I want to talk about next. So you can capture these chromosomal contacts, all chromosomal contacts in a cell, by a method that's called Hi-C. And very briefly, how that works is you cross-link strands of DNA that are in close proximity together. You cut them with a restriction enzyme. You fill the overlapping ends, the restriction fragment overhangs with biotin, and then you ligate together. And what you then have as a result is two chromosomal regions that are in close proximity now form this covalently linked hybrid molecule. So where A is linked to B, and that in the subsequent steps, then you can read out by massively parallel sequencing. So the frequency at which you detect these molecules, which of course don't exist like this in nature, you've introduced them via these molecular biology steps, is a readout for how often these sequences are in close proximity. You can capture them. So that methodology was there when we started, just about when we started to work on it, but its enormous power is also its kind of Achilles heel, because these libraries are so complex that you can't actually identify individual contacts between enhancers and promoters. And this is what we were interested in, unless you sequence to a depth that is really prohibitive cost-wise, even for well-financed labs. So we thought maybe we can, out of this ocean of interactions, that chromosomal interactions that a Hi-C library is made of, we can introduce a trick here, and we design baits against the gene promoters. This is where a gene starts. These are biotinylated RNA baits, and we use them to fish out of these in a solution hybridization reaction. We can fish out the specific interactions that we're most interested in. And for us, this was between enhancers and a gene. So we thought if we capture all the genes, we will isolate the interacting frequencies with them, and that includes the enhancers, right? And for those of you who are familiar with this technique, this kind of capture has first been introduced in exon capture, but we did quite a lot to adopt it to Hi-C libraries. So this is a screenshot of a Hi-C library where every arc here represents a chromosomal interactions. You can see there's loads of them, as you would expect from a Hi-C library. But when you zoom in, like how many are actually over this gene? In this case, it's cell1, but it could be any gene. There's quite few, and they won't be reproducible, because when you sequence a Hi-C library, unless you do it extremely and deeply, you will sequence a different tip of the iceberg every time you do it. But with promoter capture Hi-C, is what we call this technique, we capture much more interactions, and they're much more reproducible, because we reduce the complexity of this library quite dramatically. And we've done this, we've tested this extensively. So what you can see here is plotted the number of reads that involve a gene promoter in a Hi-C library in blue. So that's without the capture step, without the enrichment. It always hovers around 5%. Whereas when we apply promoter capture Hi-C to this, you can see we go up, we increase the number of promoter reads by about a factor of 15, whereas the theoretical maximum would be 20. So we're quite happy with the enrichment. The first test that we did very early on, this is now published 9 years ago now, is to check whether what we captured actually looked like enhancers. And for that, we looked at the histone marks. Enhancers are characterized by specific histone marks. Post-translational histone modifications, I'll go into that in more detail later. So what we did here is we grouped the promoters from not expressed to highly expressed along the y-axis, and then plotted the histone modifications in the respective promoter interacting regions, right? And what we saw was that the more highly expressed you are, the more likely you are to interact with a region that carries the two enhancer marks depicted in red here, H3K4 monomethylation and H3K27 acetylation. Also carry other active histone marks, but that was good. So that gave us confidence what we're actually capturing were enhancers. So we had developed this technology out of pure academic interest. I wanted to understand. I was working in Peter Fraser's lab at the time as a postdoc, and we wanted to understand, how are genes regulated, sometimes over large genomic distances, and the information we needed to ask this question, the linking the genes to their appropriate enhancers just wasn't there on a genome-wide scale, and we could do it now in one experiment. But we soon realized that there was a lot of interest in our technology from the GWAS community, and GWAS stands for Genome-Wide Association Studies. And the reason why they were so interested is depicted in this pie chart. So genome-wide association studies map genetic variants that are associated with increased disease risk or different, adverse disease progression, things like this. And not for all kinds of traits, hundreds of studies, thousands actually by now. And this is plotted over all of them in, a while ago, but the percentages haven't changed yet, even though the number have changed. What they found is that, contrary to the expectations of when these studies started originally, is that only a relatively small amount of these disease-associated genetic variants across the human population were actually located within the coding region, within protein coding regions. So you can see here, it's about 5%, lower than, higher than by chance, because by chance the genomic space that these regions occupy is about 2%, so it's enriched. Nonetheless, it left 95% of the GWAS hits in the non-coding region, right? In the outside of what encodes a protein. And further, these regions mapped to DNA, so to accessible chromatin, which is a hallmark of enhancers. So it looks like, we can't be sure for every single one of them, but the majority of these regions certainly look like enhancers. And the model then is that genetic variation in enhancers drives subtle expression changes of the genes that those regions control. Remember, they act like switches or kind of tunes for gene expression levels. And that increases your risk of developing, of acquiring a specific disease or responding to treatments or several indications. So that then led to the formation of the spin out of the company. We realized that that was a bigger question than we could tackle in our academic settings. Also, I don't have a disease background, so it was really we wanted to use this technology to really advance human healthcare, and we realized we couldn't do it academically. So we founded Enhanc3D Genomics in 2020, just before the pandemic. It was an interesting time, but it's gone really well. We're a great team now of over 20. And what we tried to do is they've renamed Promoter Capture High-C into Enhanc3D, which is more catchy, to unlock this potential of this technology to improve human health. And I'll go quite quickly through this, but what we aim to do is at least 3 strands, maybe more, but I'll focus on these. The first is novel disease target gene discovery. Remember the sonic hedgehog in lymphoid-specific enhancer example, where by assigning this enhancer to the nearest gene, which is what people have the nearest on the linear genomic map, which is what people have done by default over years, you would have wrongly assigned it, right? You would have assigned it to the wrong target genes. But by taking the 3D genome information into account, we can identify actually genes that these regions, these putative enhancers, interact with and regulate. And this we sometimes call this de-orphanization of GWAS hits. We can make sense of this GWAS data. The second is that we're exploring is if we can identify 3D genome biomarkers for patient stratification, personalized medicine, or do specific 3D contacts in specific cell types. Can they be used as biomarkers, essentially? And the third strand that I want to highlight here is the cell type prioritization in disease. What GWAS gives you is a genetic variant that is associated with a specific indication, increased disease risk, and so on. But doesn't tell you in which cell type that occurs. But we can look at that, and because we have these profiles for many different cell types by now, we can say that it not only does it regulate this in this gene, it does so in this cell type. And I can't disclose, our top targets, of course, here for IP reasons. But I can show you a really nice example from a very impressive paper by James Lee's lab at the Crick Institute in London, and that illustrates very well these 2 of the points in the previous slide: the assigning novel disease genes to regions, disease-associated regions in the non-coding human genome, and the cell type prioritization. And what they've done, they've actually used Promoter Capture High-C data as part of their study. It's a tour de force study, very impressive study. What they've done is to use Promoter Capture High-C, and they found that this interaction between this region, so this is the disease region here in this red box, it's associated with some IBD, but also other immune disorders. It interacts specifically in specific cell types, namely macrophages and monocytes. Monocytes derived from are the precursors of macrophages, but not in B cells and D cells. You don't have this interaction. And it interacts and regulates, they've shown this by CRISPR deletion of this region, not with the neighboring genes, which is PSMG1 and BRWD1, but with ETS2, which is 290 kb away. So it makes a really nice example of the power of our technology to assign the right target genes to disease-associated regions and define the cell types in which these interactions and the target genes play a role in disease. Okay, I'll come to some recent data that I wanted to go through from the lab. This is unpublished, although hopefully not for too much longer, but as of yet it is unpublished. And that is the functional adaptation of enhancers in human pluripotent stem cells. Pluripotent stem cells are of great interest to us from a developmental biology perspective, but also for regenerative therapies. So for example, they are in the use of in cell therapies and Parkinson's disease. It's just one example. The idea is that you derive human pluripotent stem cells, you differentiate them into these target cells, so dopaminergic neurons, for example, in Parkinson's, and you can replace faulty cells in the patients with healthy cells. And so they're of great interest in terms of fascinating biology because they can give rise to all cell types in the adult human body, but also they have real implications. So it's an important cell type, real biomedical implications, so important cell type to understand. And yeah, we have 3 ongoing projects in the lab at the moment, but I'll focus on this one, functional annotation of enhanced human pluripotent stem cells. So I've talked about enhancers and kind of implied we do know perfectly what makes an enhancer, what defines an enhancer. I'm going to now retract some of that and say it's not actually true. And I hope the data I'll show you in the next couple of minutes will convince you of that. Enhancers were classically identified and characterized by reporter gene assays. So you would clone an enhancer upstream of a minimal promoter. It can be downstream, as I said, the position doesn't matter for enhancers, but in classical upstream of a minimal promoter that drives, that promoter doesn't drive gene expression by itself, but the enhancer does. And it would then, you would make mouse embryos from this, for example, and then monitor where this gene is expressed. So in this case, it's a mouse forebrain specific enhancer during mouse development. This is an E11.5 embryo. These days, we've largely replaced, these are of course pretty cumbersome assays, as you can imagine, and it's hard to do them in a real high throughput manner. We have replaced this more or less by pulling down regions that are marked by marks that have been associated with enhancer activity. And these are post-translational histone modifications. For example, histone H3, lysine 27 acetylation. That is the standard enhancer mark. Some people also just use ataxic profiles, so just open chromatin to assign enhancers. I'm not a big fan of that. I'm happy to go into that later. When you go into the literature, you'll actually find that that these methods, it's important to keep in mind that these methods not actually functional readout. They map marks on chromatin that correlate with enhancer activity. That doesn't mean they really, what they capture is an enhancer in every single case, because for that, to say that, you would need this kind of functional information, that it can really drive expression of a gene. And when you look in the literature, for example, the Vista Enhancer browser is the largest database of this kind of data. So mouse embryos, which sequences drive reported gene expression in mouse embryos. 40% of the tested putative enhancers fail to drive expression in any tissue. That could partially be explained by the fact that they are outside of their normal genomic context, but it's still quite a high number. Another study from Ian Chambers' lab a couple of years ago showed that only 11% of these regions that are marked with this mark actually drive reported gene expression in an episomal assay. I'll come to that in a minute. Again, probably lower than you'd expect. So these 2 observations would suggest that not everything that you identify as an enhancer based on the classical enhancer, active enhancer mark, HCK27 acetylation, actually function as enhancer. There's also data so we would be over-predicting enhancers by that measure. there's also data that suggests that the opposite is also true, namely that there are regions that lack this mark but function as enhancers. And I've got 2 examples here. One is called unmarked regulatory elements. This have been identified through unbiased CRISPR screens of upstream non-coding regions, upstream of key genes. The second is non-canonical enhancers in mouse ES embryonic stem cells that are marked by an acetylation mark, but it's in the body of the histone H3, not its tail. So we wanted to address this in an unbiased manner, and to that we turn to an assay which is called StarSeq for a self-transcribing active regulatory region sequencing. And what you do in StarSeq, this has been established by Alex Stark's lab in Vienna, is you clone, so we chopped up the entire genome of pluripotent stem cells, cloned it downstream of a reported gene, but upstream of a polyadylation site. And then, if you remember what I told you about the ability of enhancers to drive expression in an orientation-independent manner, they would drive their own expression, right? They would stimulate their own expression. So you can read this out in the RNA-seq data. And they function as their own barcode. So you don't need to barcode that library. You can just take the entire genomic genome. And we integrate this data for reasons that will become apparent in the next slide with, because this is an episomal assay, so you measure the ability of these sequences to have enhancer function, but that doesn't necessarily mean they are enhancers at the endogenous locus. So we integrate this with chromatin accessibility data, use a TAC-seq, promoter capture IC data. This is to link them to their target genes, the technology I've introduced you, and we measure various chromatin marks, post-translational epigenetic histone marks. And this is how the data looks. Well, so what you measure is the output. This is here in 3 replicates, the top rows, the enrichment over the input, which is in green. And what you can see here, so these are 3 regions that our algorithm would identify as enhancers. This just about here, this is a weak enhancer. The other 3 are sequences, because they're clearly enriched over the green background, they function as enhancers. But what you can also see is this first one here, for example, does not overlap with the region of accessible chromatin. So most likely, and those are the majority of regions. We're not the first to see this. Everybody who's ever done StarSeq essentially finds this. So those are likely regions that have the ability to function as enhancers in this assay, but as they're not accessible on chromatin, we believe the most likely explanation is they're repressed at the chromatin level and they don't actually function in that cell type as enhancers. So there is actually data that suggests that may not be true for all of them, but for the sake of this story, we focus on the other ones. We focus on the ones that look like this, that overlap with an ATAC-seq peak. So we know it drives enhancer activity, it drives gene expression, it has enhancer activity, and we know at the endogenous side, something's bound there, right? Because that's what an ATAC-seq peak implies. So this last category that doesn't have an ATAC-seq peak, I call chromatin repressed here. I'll ignore them for now, and I'll focus on the other 3. So the other 3 all have an ATAC-seq peak, right? They're located in accessible chromatin, and we classified them by standard means. We use the histone modifications to classify them into active. They have this active histone mark, H3K27 acetylation that I mentioned before, primed that lack this mark, and poised. Poised are ones that are inactive and kept inactive by the activity of the polycomb repressive complex, PSC1, PSC2, and they have the characteristic polycomb mark, which is H3K27 trimethylation. Those are quite well characterized. These are the classical active enhancers. And this is just to show these are genome-wide trends, because obviously these upper examples here are specific loci, but they're genome-wide trends. The class that interested us most is this primed enhancer class. And they're sometimes called, you find different names for them in the literature. They're called primed, neutral, or inactive enhancers. People mean the same thing by this, by these different classifications. It's the same classification, it's just a different name. So what the model is that these enhancers actually not active, because they lack the active enhancer mark, but they lie in waiting, and they will gain this active enhancer mark once the cells transit from pluripotent stem cells into something more specialized. Could be neuronal precursor cells, could be mesoendoderm cells, anything. And then they will gain this, and only then will it become the enhancer active. But the important thing is the model predicts they're not active enhancers. So this is what we wanted to look at. This I'll skip over this, but this just to show that the majority of these enhancers are located in intergenic and intronic regions, which is where enhancers should be located. So we considered for these primed enhancers, we considered that they are primed enhancers, and that is the model that I've just outlined to you. They may interact with their target gene promoters, but the genes would be repressed because they don't function as active enhancers yet. And the alternative explanation is that they are non-canonical enhancers, that they would actually function as bona fide enhancers. So we first looked if this primed model is true. It makes two predictions. One is the expression level of the interacting genes should be lower. They should not be expressed. And when we compared the expression level, we assigned them to by promoter capture HSC to their target genes as active, primed, and poised. In these three categories, we can see that the active one, the genes are the most highly expressed, but the primed ones are much higher than the poised. And this is also true by genomic proximity. So kind of weak support for the, they're clearly, at least some of them are expressed. Second prediction is that these regions would gain acetylation, H3K27 acetylation during lineage specification. And so they would look like, for example, here we looked at 4 different cell types, mesoendoderm, trophoblast-like cells, mesenchymal stem cells, and MPCs. So they're all derived from PSCs, from these pluripotent stem cells. And we would, if the primed model is true, you would expect to see pictures like this, that they gain H3K27 acetylation either in all of them or specifically in one of those lineages. But the majority of our primed enhancers don't. See, this is the upper level here. About 60% of them don't gain the active mark in any of those cell types. So it prompted us to really test this directly. Could they actually drive gene expression? And for this, we turned to epigenome editing with CRISPR. So Cas9-CRAB is called CRISPR-I. So it's a standard assay in enhancer biology. You recruit a repressive moiety to the enhancer, represses activity. And if that functions as an enhancer, you should see the expression levels of the target genes go down. And by and large, this is what we do, although in some levels, and in some cases, the effect is relatively mild. But in those cases where we've done it, and this here, the last one, SFRP2, is controlled. That is, this is an active enhancer, canonical enhancer. We find that, in these cases, the expression level actually does go down. And remember, we're now targeting the endogenous locus. So these are actually enhancers for these genes. We've also done the reverse experiment. So we, instead of recruiting a repressive activity to the enhancer, we recruit an activator, so VP64. And in this case, you would predict that if this functions as an enhancer, the expression level would go up of these genes. We've only tested 2 of those, but that's indeed the case, what we see here. So in one case, one of these enhancers especially grabbed our attention. I go through this quite quickly because when we targeted this, and you can see this peak here is a nice star-seq peak, overlaps with an ataxic peak, also overlaps with CTCF, a key chromosomal protein. The CRISPR system we use is inducible, so we split the cells into 2, and we have the non-induced control. You can see a nice pluripotent, this is over 4 days live cell imaging, and we can see a nice growth of a pluripotent stem cell colony. This is what they should look like. And in stark contrast, when we induce CRISPR-I against this promoter, you can see that there's lots of cell death, first of all, and also the morphology looks completely different. This is just a blow up of these last images at 4 days. I hope you can appreciate how different the cells look. Of course, one is green and one is other. So that's just a marker for that CRISPR-I was actually activated. That's not the same. That's not the main point. I'm focusing on the difference in morphology and cell numbers here. We've tested, so what Cas9-CRAB should do is it should induce like a localized island of this repressive chromatin mark, which is H3K9 trimethylation over the locus. So watch this here. This is the control. When we induce CRISPR-I is exactly what we see. So you can see this extra small island, probably like 3 kb either side of the enhancer. And so it's on target. We didn't see anything that's off target that made us. It's obviously something we were worried about with such a dramatic effect. We don't see that. I've mentioned before that this enhancer is bound by CTCF. This is again the control. When we do CRISPR-I, the CTCF binding is dramatically reduced, suggesting that CTCF may have a function in this enhancer. And we do see this effect. So this is live cell quantification data, the number of pluripotent stem cells. The APLN enhancer had an effect on the target gene expression. So we know the CRISPR-I system works, but has only very minor effect on cell viability. Whereas this enhancer, which we think controls a sulfate transporter, we're not 100% sure about this because two genes go down mildly. The other one is a nucleoporin when we do CRISPR-I. So it may regulate more than one gene. But we can see this when we do standard assays. We use 3 gRNAs simultaneously to target the enhancer. We can reproduce that with the individual gRNAs as well. So it gives us extra confidence that is true. And we do see even in the cells that survive, and they have a reduced level of 2 key pluripotency markers, NANOP and SOX2. So so far I've only told you what these enhancers lack, which is the active mark, H3K27 acetylation. We're keen to find something that defines them, positively defines them. In mouse ES cells, I've mentioned this in the introduction, there's this class of non-canonical enhancers that is marked by H3K122. So in the body of the histone H3 acetylation, we don't see much difference here between our active and primed enhancers. It's more enriched than active. It's like mildly enriched in both, but certainly can't be used to identify primed over the active enhancers. The only mark we found that can slightly do this, but it's a very mild enrichment, is over the primed is H4K12 acetylation, and we're still looking into that. You can see this example here. So finally, we asked, do sequences with this signature also exist in other cell types? And we looked at the ones that are all, the human ones are all from cancer cell types. We looked at the ones that, so we classified star-seq and ATAC plus regions into ones that have the primed enhancer mark, that have the active enhancer mark and that lack them. We can certainly find many examples that drive of sequences that lack the active enhancer mark, but drive, these are the ones in red, but drive star-seq expression. So we haven't tested those functionally with CRISPR as we have in our pluripotent stem cells, but that suggests that these may be widespread. Yeah, and I'll quickly end on a model of enhancer promoter specificity. This summarizes a lot of data in the literature. How do I think? So this goes back to one of the first slides. How do enhancers find their target genes in nuclear space? And I've broken it down, or we've broken it down, and Peter and I wrote this review 5 years ago or so, into 3 steps. The first is selecting. Out of there's probably 1 million or so enhancers in the human genome, but in every cell type, only a subset of those will be active. So they need to be selected somehow. That's a big question of how this is done. It could be likely involves pioneer transcription factors, other transcription factors, histone modifying enzymes, chromatin remodelers, all kinds of things. The second step for me is what I call facilitating. So there are mechanisms that make initially random enhancer promoter contacts more likely, and that includes transcription factories, phase-separated condensates, and probably also topologically associating domains and compartments. And the third step is then for me that specifying, so it is a kind of a key lock model, that specific interactions occur between the proteins that are bound at enhancers and promoters that mediate these interactions. And yeah, this is just a run through of all the things I've talked to you about. I hope this is clear. I'll end with thanking. So this is my team here at the Bagram Institute. The StarSeq was a big collaboration with Ufuk Nastugan's lab, the University of Göttingen, and here on the right is all the fantastic people at Enhanc3D Genomics and the others. We have great support here at Babraham from facilities, and we've also interacted with Pradeep Madhapura at the Blizzard Institute in London, Queen Mary University in London. And with that, I'll acknowledge the funders as well, and I'll be happy to take questions. Thank you.
Excellent. Thank you so much, Dr. Schoenfelder, for that fascinating presentation. And we've already had some excellent questions come in, so I'm going to start with the first one right away. So what are the key nuclear proteins that organize the human genome in 3 dimensions?
Yeah, that's a great question. So in terms of the architecture of proteins, that's quite well known. So the main 2 players is the cohesin complex and CTCF, and they mediate together what's called loop extrusion. So the cohesin complex kind of binds to regions and then pulls the DNA out. It's kind of this ring-formed complex, and until it stops, finds CTCF sites in convergent orientation that stop its activity. And that is what kind of makes loops into genome. For example, TATs, topologically associating domains, are formed like that. But that doesn't explain how enhancer promoter loops are formed. And that is still an open question. I doubt that there's one protein. For years, people have hunted this one protein that mediates enhancer promoter context. I doubt that exists. I think it's combinations of proteins that sit on the enhancer and on the promoter and that are compatible and that mediate these interactions.
Excellent. Thank you so much. Another really interesting question we've got here is, how is the 3D organization of the human genome perturbed in disease?
That's a great question. There's multiple mechanisms. There's excellent reviews out on that. It's an ever-growing list of enhanceropathies, as it's called. So diseases that are caused by mutations in enhancers or perturbed enhancer function. It can be that enhancers access genes they shouldn't access, otherwise they are kind of normal, and that drives the aberrant expression of these genes during development. That can lead to developmental malformation. Stefan Mundloh's lab in Berlin has several key papers on that, for example. The second big example is that the enhancer is faulty, so a mutation inactivates its activity. And again, multiple examples in cancer and developmental malformations.
Lovely. Thank you so much, Dr. Schoenfelder. Unfortunately, I think that's all we have time for today. So we appreciate you taking the time to share your expertise with us, and any questions that we didn't have time to get to will be sent to Dr. Schoenfelder after the event. As with all the other talks in this event, this talk will now be available to view whenever you want for the next 30 days. And that just leaves me to say thank you so much again, and we'll be back soon with the next talk.
Pleasure. Thanks for having me.